everybody, it's me again, here to talk to you a little bit about cellular energetics. So let's start with bioenergetics. So basically our overview right now is I'm just going to kind of go over a little bit about what is energy and how does it mean in terms of the cell, and let's talk about a little bit about energy. So some of you guys might have remembered there was this guy Newton. He actually didn't sit under a tree and have an apple fall on him, but he did come up with a couple of theories in terms of theory of laws of thermodynamics. And his big laws of thermodynamics that he came up with, the first one, energy cannot be created or destroyed. That means everything's conserved. So, if you sit there and you're slamming on your brakes in your car, you are putting friction in there, and it's also releasing some of that energy as heat. That you're not creating any energy, you're not destroying it, you're just converting it. The second law is that energy transfer always leads to less organization. And you guys all know that just by looking at your rooms. I'm sure sometimes your rooms are not as nice and put together as it can be. If you don't put any energy into it, eventually it's going to be destroyed and less organized, and that really sums up that second law. Since energy can never be created or destroyed, and since also energy leads to disorder, that leaves us with two types of reactions that you can get. And this isn't any type of reaction. Either you can have an exergonic reaction. And I always think of exer is outside, coming out. And basically what you see is that your products have less energy than your reactants. So again, looking at this energy diagram, you can see that energy is represented on the y-axis. And so we start with a lot of energy here in the early part of the reaction, but we end up with less energy at the end. And that difference is our delta G, or change in energy state. And the reaction releases energy into the system. Another type of reaction is an endergonic reaction. Here you can see your products have much more energy than the reactants. The free energy has increased. So again, we see this a lot in things like even plants when they use carbon dioxide and water. Those are some very low energy molecules, but they end up making a very high energy product, which is sugar. Now, luckily, in your body or any systems, we don't go directly from the amount of energy in the reactants to the products, just like this, because if we did, then we'd have to worry about when these reactions would occur. So sometimes reactions are going to need a little bit of energy to get things going. You have to kind of go over what we call an activation energy. I like to use this as an example of something like a match. If you have a match sitting on in your pocket or someplace else, what you may notice is that it doesn't go off when you don't want to light it, which is a good thing. Because can you imagine having a whole bunch of matches in your pockets and then all of a sudden they're going from the reactants to products just like that, and they would just spontaneously combust at any time. So instead, what ends up happening is we need to put a little bit of energy into it so that we can get that flame to come out when we want it. And that energy usually entails a little striking or a little bit of friction that we put into our original match. And we're doing that so that we're putting a little bit of energy in so we can control when we're turning it on, so then it's a downhill right after that. And that's really important. That's that activation energy. So we can kind of control when things get turned on and turned off. Now, sometimes getting to this state over here can be very difficult. So enzymes are utilized in systems to help lower that activation energy. And that lowering of that activation energy helps for the reaction to complete to the reactants. And we're going to talk a little bit more about that when we talk more about enzymes, which is coming up right now. A little bit about enzymes. Basically, they're biological molecules that speed up reactions. They speed up reactions by lowering that activation energy that I just showed you in that last graph. And what they're really doing, they're helping to make a transition state. What do I mean by that? Well, here you have two molecules. Let's say you have molecule A, you have molecule B, they're attached. And at the end, you want to get a molecule B and C to be attached. Well, what the transition state is going to be, it's going to be a state where you see A plus B plus C all together. And that, as you can imagine, is something that's going to require a lot of energy. That's why there's such a need or such a high amount of energy to get there, because these are molecules that normally don't want to be together. They don't fit together normally. So at the end, we can get something, at the end of the reaction, we can get that A molecule by itself and eventually B and C together as a final product. In addition, enzymes do not change the energy of the starting point or the ending point of the reaction. They're very specific. They're only going to catalyze one type of reaction, and their targeted molecules are known as substrates. And one thing that I did not put in here that I don't remember if I mentioned, but it's also kind of important too, is that enzymes also don't change in the reaction. If you notice here, my reactants changed. Oops, won't do that. My reactants changed. My transition state changed, and my products changed, but the enzymes don't change at all. Now, enzymes' jobs are going to help to bring this transition state about, and I notice that I have a small little typo, by helping the substrate into position. So again, kind of looking what I talked about before, here's your substrate. It puts it into a position into the active site. And basically, this enzyme now can change shape in such a way that maybe it puts a little tension, so it breaks these bonds. And by breaking these bonds, it ends up making two new products or something to the effect. And that's all going to occur at that very important part, which is called the active site, right over there. So just as a quick review, remember, enzymes are going to increase the rate of reaction by lowering the reaction's activation energy. It's going to form a temporary enzyme substrate complex. And it's going to remain unaffected by the reaction. Enzymes don't change the reaction. They don't make the reaction occur that would otherwise not occur at all. Now, I have heard people say that enzymes have a lock and key mechanism. In other words, they're so specific that enzymes are going to be like a key like the lock that fits perfectly with the key that is the substrate and eventually going to give you the product or the reactants. But that's actually found not to occur anymore. What they have found, scientists have found, is that it's really doing what's called an induced fit reaction. Basically, it's that they don't fit together really perfectly, the enzymes and the substrate. And in fact, once the substrate goes in, it fits fairly well, but the enzyme's actually going to change shape a little bit. And why is it changing shape? Remember those bonds that we've talked about in those proteins? We have the 3D structure of the proteins have those hydrogen bonds, ionic bonds, hydrophobic bonds. They have the van der Waals forces. All of those disulfide bonds, all those bonds are going to come to play to do an induced fit or change the enzyme just enough to accommodate the shape of the substrate. Side note, what you guys can't see right now is that as I'm talking to you, I'm using my hands to describe everything, and I'm kind of laughing at myself for doing that. All right, now how many of you guys have heard that it's really, really important to get your vitamins, to get your minerals, and stuff like that? Well, do you know why those are important? The biggest reason why vitamins and minerals and extra ions that you might be getting from your diet or supplements are important is because enzymes don't work alone. Sometimes enzymes need other things to help them work, and those things can be your cofactors. Cofactors can be broken down into either metal ions or coenzymes. And basically, they're either organic molecules, such as that can bind with the protein or something and help it work better. Like your vitamins are going to be organic coenzymes. They help the enzyme work correctly. Or you can have things like iron or magnesium. We know iron is very important to get hemoglobin to bind oxygen. And so these things or molecules are important to help enzymes work, and they don't work alone in that sense. Now, these are things I feel like I don't need to go over with you guys because I'm almost certain you've learned it really, really well in your regular bio class that you had before you came to AP Bio. But basically, some of you guys know that things like temperature is going to affect the rate of reaction. And again, as you start to cook things up and add fire or heat to your reaction, that's my little fire going, what's going to happen to your proteins is they're going to start to unravel. And we've seen this example if we've cooked a nice egg or something. You might have noticed that the meat or whatever you're cooking changes form even, changes shape. So I'm going to make a little egg here. Here's your little egg cooking in your pot. No, it's really hard to show white, so I'm going to show it like that. It changes shape as you add the heat, and the reason why it's changing shape as you add heat is because you are denaturing the enzyme. Remember, enzymes are proteins, and they're losing shape. Too much heat's going to damage your enzyme. Sometimes you can increase it a little bit to increase the rate of reaction, but you have to worry about how high you get. Too hot, you're going to lose that three-dimensional shape, and you're going to inactivate it. The other thing that affects enzymes is that pH scale, so 0 to 14, where you have acid at the 0, 7's neutral, and we're basic at 14. And again, some proteins like to work at most of our body likes to work at 7. We do know that things like our acids, our stomach acid is around a pH of 2, so we sequester that. So those enzymes work the best at pH of 2. And other things that can regulate your enzymes are inhibitors. And we have different types of inhibitors. We have the competitive inhibitors. They're going to bind to your active site. What's interesting with a competitive inhibitor is that, let's say you have, here's your substrate, here's your enzyme. I'm going to make it really easy, something like that. And you have substrate that fits in there, big circles. And then I'm going to make a weird inhibitor. Let's do something like that. That's circular but has a weird shape to it. What's interesting with competitive inhibitors is that the more substrate you have, the more chance that that's going to fit into the enzyme and kind of block out your competitive inhibitors. So you can actually avoid or change the effect of a competitive inhibitor by adding more substrate, and you're going to change the outcome of the reaction, whether it can occur or not. That's not the case with your non-competitive inhibitors. Your non-competitive inhibitors are going to bind somewhere that is not the active site, so what we call the allosteric site. And an allosteric site is just basically a place that's not the active site. And by binding to this site, you're actually going to change the shape of the active site. Again, we're dealing with bonds. And put it in there. And by changing the shape of that active site, it doesn't matter how much substrate you have, it can't get in, and you've stopped the rate of reaction of that case. By the way, sometimes when looking at drug production, what you want to do is you want to find either a competitive inhibitor or a non-competitive inhibitor to affect the transmission or the production of a protein that might not be good, or you might have too much of, or something to that effect. So these are places that we do look at in terms of drug discovery. Okay, so again, when we talk about energy, we usually talk about in the cell, photosynthesis, and cellular respiration. So let's start first with photosynthesis. Okay, so what is photosynthesis? Hopefully you guys all understand and know that it's basically light energy is converted to chemical energy. So you have your lovely little plant is going to get the sunlight, and everyone always draws the sun in a corner for some reason. And it's going to pick up, the sunlight's going to be picked up by the plant. We have its little leaf. It's taking that energy, and we're going to make sugar out of that sunlight. We're also going to take in some CO2 and water to help convert that into the sugar. And it's divided into two phases. Those two phases are the light-dependent reaction and the light-independent reaction. And just as a quick review of some vocabulary, when we talk about photons, we're talking about packets of light, and the job of those photons is to really activate the chlorophyll and excite those electrons. So again, let's go over that light reaction. Those photons, again, those packets of light, are going to hit photosystem 2. And I just realized that I got this awesome figure from some book that does not have English as its first language, because it's spelled a couple things a little different. But it shows you how biology is such a universal language. It's almost the same in any language. Now, when that light hits the photosystem 2, it's going to excite the electrons. And those electrons are going to, again, get passed along to eventually it gets passed to a primary acceptor that's going to take it to photosystem 2. So it's going to go through an ETC, electron transport chain, going to get picked up as an electron acceptor by photosystem 2. And now that I look at it, this is also, oh, I said it wrong. Sorry, photosystem 1. My bad, said it wrong. And then the other thing too is that once we transfer this electron to photosystem 1, what's going to happen is that we're going to lose the electron here. So we have to replenish that, and that electron is going to get replenished by hydrolysis of water, which is basically going to break down that water to oxygen. This is the oxygen that leaves the cell, so it's going to leave the cell, leave the plant. And this is the water that we also use to give to our plant. That we use in that sense. The biggest important thing is that this transfer of all these electrons is going to pump hydrogen into the lumen of the cell. Okay, and this helps build up a proton gradient. So you get all these hydrogens going to build up on the inside, and then eventually those electrons or hydrogens have to go back to the stroma, and by doing so, it's going to help power ATP synthase to help make ATP. So the real big thing about the light reaction is a couple things to point out is we are going to make some ATP, and that ATP is going to be used later as energy to help make sugar. So now let's turn to the dark side, or the light-independent reaction. I have to put my own little nerd joke in there for you guys. But basically, what the light-independent reaction is going to occur, it doesn't have to have sunlight for it to occur. It's going to use those products that we just made, that ATP plus the electron carrier, the NADPH. We're going to utilize those to help make sugar. Okay? That CO2 that was brought into the plant is also going to be used to go through carbon fixation, which basically is where you're taking carbon from the air and you're converting it into carbohydrates. And all of this occurs in the stroma of the plant. So as just brief overview, what ends up happening and where it goes is a great little chart that kind of shows you the stage of the photosynthesis, where it's located, what comes in, and what comes out. And again, something that you can definitely utilize or keep available for you as you take that AP exam. You might have to work it in some way or something like that, but those are just good ideas to keep for you as in front of you as you reference it for the AP exam. The last part of the energetics unit is cellular respiration. Okay, and basically it's to take that sugar and oxygen and help make a ATP. We're going to make some water, and we're going to make also carbon dioxide as we do our exercises. There are 4 stages of cellular respiration. There is glycolysis, the formation of the acetyl-CoA, the Krebs, also known as the citric acid cycle, and then finally oxidative phosphorylation, which basically consists of the electron transport chain and chemiosmosis. So what is glycolysis? Glycolysis is basically splitting glucose to pyruvate. It's shown here in this process here. You are going to need ATP to start the process or to basically prime it. So you are going to utilize ATP in this process early on. So at the end, you end up making 4 ATPs, but because you use 2 early on, the net gain is 2 ATPs. The biggest thing you're also making are electron carriers, NADH, which is going to be used later for the electron transport chain. And all of this occurs in the cytoplasm, and at the end, you end up getting 2 pyruvic acids. Those pyruvic acids are then going to be utilized as it goes into the mitochondria to form acetyl-CoA. And again, what happens is we're going to convert it using the enzyme acetyl-coenzyme A. More NADH is made. Again, those are really important. And CO2 is released, and this is again released as you exhale out. Once you get into the mitochondria, into the matrix is that middle part of the mitochondria. The acetyl-CoA is going to join up with a 4-carbon molecule called oxaloacetate. And acetyl-CoA has 2 carbons in here. So that 4 carbon and the 2 carbon, hopefully you can do a little math, will equal 6 carbons altogether. And then as it goes through that cycle, that 6-carbon molecule is eventually going to transform back to a 4-carbon molecule, so it can rejoin with another acetyl-CoA. But all that reforming is going to eventually release CO2, it's going to release some ATP, it's going to release more electron carriers, which is really important, so that we can, again, drive that electron transport chain. The last part of cellular respiration is oxidative phosphorylation. And this is where those electron carriers, so again, your NADH and your FADH, are going to drop off those electrons. Those electrons are going to go through that ETC, or the electron transport chain. Your final electron acceptor is oxygen, and that's going to help make H2O. But again, the key point here is we're increasing that H gradient, the hydrogen gradient, which is going to be used to pump down to through the ATP synthase, which is going to make lots of ATP through this. So you end up through this whole process making anywhere from about 30 to 38 ATP molecules from aerobic or cellular respiration. Now sometimes you don't have enough oxygen, because remember, oxygen is that final electron acceptor, and if you don't have enough oxygen, you cannot go through aerobic respiration. So what you're going to have to do is you're going to have to go through an anaerobic respiration. So the ETC is not going to work at this point. You're still going to have glycolysis occurring, but now you're making only 2 ATPs and not 30. The goal of this is to help recycle the NADH. And depending on the organism, either you're going to make lactic acid, which is done by humans, or you're going to make ethanol, which is going to be done by yeast, is that byproduct from this. Well, I hope this gave you a good overview of energetics, and let me know if you need anything else.